We're going to take a look at the histology slides that we need to know for the endocrines as well as the white blood cells. And so here we have an image of the anterior pituitary. Anterior pituitary has three different kinds of cells in it. And so we have acidophils, basophils, and chromophobes. And the cells themselves kind of go along a general pattern. And so we'll start with the chromophobes here. Chromophobes are essentially, as the name implies, afraid of color. And so here we have a chromophobe. And here's another chromophobe in there. And so their cytoplasm doesn't really take up any stain, so they don't have any coloring to them. Versus an acidophil is going to stain on the red side in terms of the cytoplasm there. So all of the cytoplasm portion is a redder color. And then we have basophils. And so basophils are going to have a blue kind of a hue to the cytoplasm. Here's a couple more basophils. So basophils blue, acidophils red, chromophobes no color to them, and we have our anterior pituitary. The posterior pituitary, as we've seen, is made up primarily of nervous tissue. So it has a large number of nervous fibers running through it, and so kind of all of the squiggly components that are in here are all of the nerve fibers that are running through the pituitary gland itself. It does have cells. Those cells are the pituocytes inside there. And things like blood vessels, you don't need to be able to identify on the slides themselves. So here we have a large blood vessel, but we don't need to know that that's what that is. Next up, we have the adrenal glands. And so the adrenal gland has two major portions to it. It has the medulla, and then it has the cortex. And there's our dividing line between the two that are there. And so when we look at them, the medulla and the cortex, so here we have the cortex, and here we have medulla on there. And within those two, when we look at the different components that are there, we can see that the medulla is basically all of the same tissue when we look at it. And so in a sense, we kind of have the same structures that are there all throughout the medulla itself. And it looks pretty much consistent from top to bottom through that medulla that is there. For the cortex, we can divide the cortex up into three different portions. And then it also has a little bit of a capsule on the top here. So part of this part that is kind of coming off of there is a portion of the capsule that sits on the top of the tissue itself. And so we're going to have three different what we call zona. And so we have zona glomerulosa, zona fasciculata, and zona reticularis. And so it kind of goes in GFR. So we have our glomerulosa, fasciculata, and reticularis for the three different layers that are there. Zona glomerulosa tends to have kind of, as far as its appearance to it goes, it has kind of more round looking structures. The fasciculata tends to be kind of in columns. So we can kind of see a column nature there. And then zona reticularis has kind of just a mixture of the two, but one of the other components that we can generally go by as far as identification is that we kind of go from a dark to a lighter to a dark and then to a darker again when we go from the outside. So the capsule on the very outside, we then have zona glomerulosa, our actual first true layer in a sense, and then zona fasciculata, zona reticularis, and then the medulla. And so we have the adrenal medulla on there. And so each one of those is going to be functioning in producing a different type of general class of hormones. Next up, we have the pancreas. And so here we have the pancreas, and you'll remember that the pancreas is almost entirely exocrine in nature. So these are all exocrine cells in terms of the tissues that are here. And then we have these little kind of islands. These are the pancreatic islets or the islets of Langerhans. And so those tissues that are there within that is going to allow us to have the production of our endocrine side. So the endocrine is kind of few and far between as far as the tissues go. We'll go to a little close-up here of one of those islets. And now stained a little bit differently, now we can see the differences in the cells that are actually inside the islet itself. And so once again, we've got blue cells and we've got red cells. And so this is going to go along the same kind of a naming scheme in a sense. Those that are blue, here's a blue color here, are beta cells. And those that are red are alpha. And so alpha cells stain red in their cytoplasm, beta cells stain blue in their cytoplasm, but they have generally the same kind of a scheme that goes along there. And again, different hormones being produced from each one in terms of our insulin and our glucagon. Different picture. This one here allows us to kind of think about the thought process that not all slides look exactly alike, but the general pattern is there. So once again, we have an islet, pretty well defined in there in terms of the overall islet itself. And then we once again have kind of red staining cells. And then we have blue staining cells, or in this case, it's almost like more of a dark purple in terms of that. And so we once again have our betas, and then we have our alphas. Here we have the thyroid gland. Thyroid gland has some different structures to it. And so the big open spaces are the follicles. So those follicles are filled with the colloid that is there. Surrounding the follicle, we have individual cells. So they go all the way around there. And those are our follicular cells, or a follicle cell. In between the cells that are, in a sense, not touching an actual follicle, these are our parafollicular cells, or our C cells. So those are the cells that are not involved in the production of thyroid hormone, just the follicle cells themselves are. And so it kind of looks like adipose to a certain extent, kind of looks like lung a little bit as well. So not to be mistaken with either of those tissues. Next up, we have the parathyroid gland. And so parathyroid gland is sitting on the back of the thyroid gland itself. So they're just posterior to it. Here we have oxyphil cells. And so the oxyphil cells are the larger kind of clear looking cytoplasm to the cells that are there. And so those cells are going to have essentially, they're almost like a chromophobe as far as the cell goes. So they don't really have very much as far as the stain in the cytoplasm that is there. So we don't see that all that much. And then the other cells that we have there are the chief cells. And so the chief cells are the smaller looking cells, and they're also the more abundant cells that are there. And so two different kinds of cells within the tissue itself. Kind of the oxyphil cells, not really a well-known function for those cells, versus the chief cells are the ones that are producing the hormone for us in terms of parathyroid. Once again, just a different picture showing us kind of the differences that we can see there. These larger cells are those oxyphil cells, and then over on the side here we have all of our chief cells inside that parathyroid gland. Here we have now the thymus. The thymus has two different components to it as well, and so we have a cortex and we have a medulla once again. So similar to what we had for the adrenal gland. And so we have the cortex here out on the outer aspect and the medulla in the inside portion there, kind of surrounding it. It also tends to kind of look like the pancreas did, except that in this case, since the medulla is in a sense kind of the center portion, it's also a much larger component. So whereas the islets of Langerhans in the pancreas didn't really have a whole lot as far as numbers of the islets. Here they make up a relatively large component to them. That's there, and so the different tissues that are there within change kind of the components that are there for us to be able to see on there. Depending upon whether we're looking at a young thymus gland or an older thymus gland, is going to kind of change the lobules and kind of the space in the septum in between. It's one of the unique things as far as the thymus that it does degrade over time. So we do see some changes within the cortex and the medulla as they start to kind of shrink as we age. And then the spaces of the septum are going to become much larger as we age as well. And so we can see those changes that are there. And so this one's a relatively young specimen. This one being a little bit older. So this particular specimen has a lot larger septum that are there to be seen. We can still see the cortex and the medulla and the differences between them, but we can definitely see how the septum has grown in size in between the tissues themselves. Last thing to talk about are the white blood cells. And so the white blood cells are the component of our immune system that's going to allow us to attack whatever it happens to be that is kind of coming after us, so to speak, on those. And so this slide here kind of gives us a good way to remember the names of our white blood cells, as well as the relative percentage that we have. And so the mnemonic there, never let my engine blow, 60-30-8-3-0, is the first letter of the saying goes with the first letter of each of the white blood cells on those. And so we have the different components that are there for each one of those. And when we do a blood sample and we pull a WBC, we're going to look at not just at times the numbers of cells that are there in terms of the total number. We're also going to want to do a differential to figure out, in a sense, how many exact of each type do they actually have in that particular function. And so how many of the individual cells do they have, which can then kind of give us a idea of what potentially is trying to, in a sense, attack the person. And so what kind of an infection are they potentially fighting off based upon those white blood cells that they have elevated within the body itself. And so each one's going to have kind of a little bit different in terms of the thing that they need to fight off on those. But the relative percentage in every single lab is going to be a little bit different. Every book's going to be slightly different in terms of the numbers, but these are generalities. So 60% of the cells are going to be neutrophils, 30% lymphocytes, 8% monocytes, 3% eosinophils, and essentially 0% basophils. And that doesn't mean that we literally have no basophils. When you do a white cell differential count, you're going to look at the individual cells and count 100 cells. And as you count those 100 cells, essentially the old way would be to just kind of make a tally as you go along and kind of keep on tallying them up as you keep counting them. And then we see a couple more, etc. On those. And so those different white blood cells are going to have different numbers as we count those 100 cells that are in that particular part of the slide itself. And then depending upon the type of infection that we're having, the different levels will see kind of a variation in terms of those numbers. But if you know the 60-30-8-3-0, you have a pretty good idea of where those should be. In total, oftentimes with basophils, you may find 1 or even 2 in a sample. Same thing with eosinophils, you may find 4 or 5 within a sample, even maybe 6 or so. And so those numbers aren't strictly 60-30-8-3-0. They're within a mild range of them. So we'll start off looking at the neutrophils. And so neutrophils have a multi-lobed nucleus to them. So we kind of have, it almost appears like there's 3 different lobes inside here. So it has a very distinctive appearance to the individual nuclei. And that's going to be one of the major factors when it comes to differentiating the white blood cells is what does the nucleus look like? From there, we look at kind of the size of the cell. And so this one is 2 to 3 times the size of a red blood cell as well. Another neutrophil, or another couple of neutrophils, and we have those individual cells there, relatively large. And again, they've got those multi-lobed nuclei inside of there. So many nuclei inside, or not many nuclei, but the nucleus itself isn't just a big, nice, round circle inside the cell. It has many different lobes to them. Next up, we have a lymphocyte. And so lymphocyte always kind of reminds me of like the moon. So lymphocyte, lunar kind of a thing. It almost looks like a crescent moon type of an appearance to it, almost as if part of it was in shadow here, and all we're able to see in a sense is kind of this portion of the moon that's there. Lymphocytes themselves are relatively small comparatively in terms of white blood cells. They're going to have a similar appearance to them in terms of their roundness and their largeness to red blood cells. They're going to be close in size to them, a little bit larger than they are. But the big hallmark here is that the nucleus basically takes up the whole cell. And so when we're looking at these here, that large dark nucleus that's inside here almost occludes all of the cytoplasm. So there's basically nothing left there to see. Even sometimes when we see a lymphocyte, we're going to see that basically the whole inside of that cell appears to be dark. And that's just because of the way that it was rotated. That nucleus basically took up the entire cell at that point. So we go from our lymphocyte to a monocyte. Monocytes, always kind of like monolithic, just huge in terms of their size. They tend to have a bilobed nucleus that is there. And that nucleus, if we were to kind of turn this picture on its side, it kind of looks like a U. And so it has kind of a U-shaped to it. It can also have a bit of almost like a kidney bean shape appearance to it. So it can have kind of a, and that's not the best kidney bean shape appearance to it, but that I can draw. But that's kind of what we have here in the picture as well, is that it kind of has that U shape or bean shaped appearance to the nucleus that's there. And again, the cell itself is relatively large. We can get 3 or 4 red blood cells inside that one cell. A little bit different one. This one is the monocyte as well. And so we have a nice U shape to that nucleus. Again, a relatively large cell that is there. And this generates the size of the cell. Eosinophils. Eosinophils are almost always red in appearance. And so their nuclei, their cytoplasm stains very red. And so we see a red appearance to the cells themselves. They're very distinct when you're looking at kind of a large span of a blood smear. Eosinophils do tend to stick out, but at the same time, they are relatively rare. And so it does take a bit of looking around a slide to be able to see those eosinophils that are there for those. And so that then adds to our last cells. So we have our eosinophil, and then we have our basophil. Basophils are very dark staining. They have a large number of these dark purple granules inside them. So many dark purple staining granules inside the cytoplasm. We can't really see much of a nucleus there. Can't really see a nucleus at all when we see that there. The big thing is that it looks like kind of a bumpy, dark purple cell is almost what they appear to look like. And then we have one more here. And again, we can see the dark staining granules that are inside there. Lots of histamine inside these cells. All those little bumpy kind of appearance to the cells themselves makes it a very distinctive cell in terms of its shape. And so you need to be able to identify the individual endocrine cells, the endocrine tissues, and then the cells that may appear on those endocrine tissues. And then you need to be able to identify the white blood cells, be able to give the relative percentage that we may find them in the blood, as well as what type of infection or what is going to bring those white cells up if we have a particular infection. So if we have, say, a basophil here, what is going to make that basophil become elevated in its numbers? What kind of a problem with the immune system is going to cause that to occur? And that will do it for our endocrine and blood cells.